People have been changing their looks for forever, but how can one change a human being and affect all the generations to come by just switching some molecules called DNA? Hi everyone, good afternoon. I'm Ecem. I'm a sophomore at Chemical Engineering Department, and today I will be talking about the 2 uses of CRISPR genome engineering. Here's my outline. The presentation today is divided into 4 parts. First, I'll give a description of CRISPR, and then I'll give some background information, and finally, I'll give the usage of 2 uses of CRISPR, which are health and farming and food, respectively. Let's start by explaining what CRISPR is. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A big mouthful of words, am I right? Unless you're a student in biology department, this description may confuse you, as it should. But don't worry, it's much more easier to understand how it works than to understand the description of it. Imagine Cas9 enzyme as a very precise scissor to make breaks in DNA. Thanks to CRISPR methods, scientists can edit the genome of living organisms by adding new fragments of DNA or replace a disease-causing mutation with a healthy sequence, or make other modifications, or even knock out a gene entirely. Let's move on to the artifacts of this victory. As you can see in the figure 1 in Alexander Heinel's 2020 picture, biochemist Dr. Jennifer Doudna collaborated with microbiologist Emmanuel Carpentier on CRISPR methods. Their work has been recognized and they won the Nobel Prize in Chemistry in 2020. Furthermore, Dr. Doudna established Innovative Genomics Institute, IGI, to search more of gene editing techniques and to make it more accessible and affordable to everyone. In Ian Taylor's 2021 article, CRISPR, a guide to the health revolution that will define the 21st century, published in Popular Science Turkey, Doudna and Carpentier advised the use of CRISPR to switch off genes that lead to diseases, to change the genetic inheritance, to prevent the diseases even before facing any symptoms, or to eliminate the risk of malaria by simply editing the infectious mosquitoes. Now, I want to introduce you Victoria Gray. She's on the left here, who is one of the first people in the world to be treated with a CRISPR-based therapy. She was born with sickle cell anemia, which is a disorder in which there aren't enough red blood cells to carry oxygen. She was on very severe painkillers and she needs regular blood transfusions. But ever since her edited cells with a gene that can produce a protein called fetal hemoglobin, which facilitates to diminish the symptoms of sickle cell anemia, are inserted in her body, she doesn't need any medication or any blood transfusions at all. However, this technology may raise some ethical questions too, like what happens if scientists start to just modify the babies or embryos? Well, that's exactly what happened. CRISPR twins were born in November 2018 in China. Biophysicist Dr. Zhuang He has delivered the first babies whose DNAs were altered. He extinguished the CCR5 gene. In this way, babies are protected from HIV virus. His work will be passed along to the following generations. However, he didn't get away with this. He was locked up for 3 years and the court fined $430,000. Although Dr. Brad Ringenstein on the right, who is the director of IGI, is shooting for the stars by saying that all diseases on the map. For now, we have dementia, Alzheimer, blindness, cardiac diseases, transitory amyloidosis, and AIDS. In May 2021, a biotechnology company launched a set of trials and observed a 59% decrease in bad cholesterol. Also, in June 2021, an experiment with human trials reported a decline in protein levels in transthyretin amyloidosis patients, which is a rare inherited abnormality in protein levels in tissues and organs. Another company called Editas Medicine is working on a CRISPR therapy for Leber congenital amaurosis, which is the most common cause of childhood blindness. They aim to restore the function of light-sensitive cells before children lose sight completely. As for AIDS, scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. Now, the second point I want to mention is the usage of CRISPR in farming and food. As you can see here in the figure 2, selective breeding is a technique that people choose parents to breed together and produce offsprings with more desirable characteristics, which is very similar to CRISPR. Only CRISPR is faster, safer, and more creative because it enables to add new traits to the DNA. With CRISPR methods, we have rice with lots of vitamin A, tomatoes with more lycopene, which is a very precious antioxidant, and broccoli with increased amount of cancer-fighting compound, as you can see here in the picture. And wheat without gluten, non-allergenic eggs and peanuts, and finally pigs that have more muscle and less fat. Following this, scientists can put gene variants or new genes into plants to protect them from pests, which are insects that eat the leaves, fruits, or any other part of the plants. You may recognize the word pests from pesticides, which is a chemical that kills pests. So why do we go into so much trouble and like edit the genes and add new traits, whereas we can just spray the pesticides all over the place? That's because pesticides have some very serious downsides, like polluting water and soil and imperiling farmers. Sometimes they can hurt food insects like bees too. Gene editing could help scientists to grow crops faster, bigger, and more in amount, and more nutritious. CRISPR helps the earth by growing crops that need less water, fertilizer, or pesticides. In addition, gene editing can be used to fight with the conditions of unusual weather by producing rice that can survive floods, or tomatoes that can survive droughts, or stock forming cows, chickens, and turkeys that can tolerate heat. Finally, let's summarize the 2 points I made. CRISPR genome engineering is used to break and edit DNA in a very precise and accurate way. As you can see here in the picture, we can break down the DNA in very close sites. This quality can improve the health of people, plants, and animals. By editing their DNAs to gain more efficiency from plants and animals and improve the quality of sick people by improve the life quality of sick people. Hopes are high that CRISPR-Cas9 technology will soon target and destroy diseases such as cancer and AIDS, and even target genes that are associated with mental illnesses. Thank you so much for listening to me. Do you have any questions? Here is my reference list and my figure of this.